So welcome to the Outside Salon. And thanks for coming despite the weather. I know that this has been challenging for some people, and I know that some people have been coming from far long away. So I'm very happy that you guys were able to make it. So today we have a special outside salon. It's called From Cell to Canvas, Creative Exploration of the Microscopic, and it's been specially curated by Mellissa Fisher, who is an artist from UK, and she is joining us today at a very particular time of the year. Thanks for coming in the winter. Yeah, she likes it. So, first, before I start, for those people who have never been to the Outside Salon is art and science collective. Sorry, I'm tired. It's Friday, and we've been trying to establish some collaboration between artists and scientists, and what better place than doing it in a scientific institution such as the Fields Institute. So we're very grateful that the Fields Institute has decided to host us. And sporadically we also have some sponsors from the sciences, so the scientific community is definitely a big supporter of this, and I wanted to go further and have artists and scientists to collaborate even further to achieve some common ground and some common research together, which is something that is our goal. I say our because Outside Salon is two people mainly. It's me, Roberta Buiani, and Stephen Morris, who is here, who is the science connection. I am the art connection, I guess. So, before we start this fantastic event, which will feature 5 artists, I would like to make I guess usual but very meaningful land acknowledgement, and I would like to remind you that despite the fact that we are in a scientific institution and usually scientific institutions are considered neutral, we are still on sacred land. And University of Toronto has been on sacred land for a long time. They actually occupied it, and this sacred land has been here for like 15,000 years, and there have been the Huron-Wendat, the Petun First Nation, the Seneca, and the Mississaugas of the Credit River. So we should be grateful and give thanks and keep this in mind while we are having fun and we are talking about art and science. So without further ado, I am going to introduce this event. You already read what it is about. This is a very diverse event. The artists who are presenting tonight are engaging with a number of different materials and a number of different topics concerning the arts and the sciences. And I would like to introduce you to the artists. So the artists in the poster are in alphabetical order, but tonight the artists will go in the following order. So the first one will be Mellissa Fisher, followed by Robyn Crouch, then Shavon Madden, Tosca Teran, and Julia Krolik. So you read their bios in the invite that you probably received. That's why you're here. So the first one is Mellissa Fisher. I'm just saying that she's coming all the way from the UK. just. And so you present yourself, I guess.
Cool.
Okay.
That's a really nice necklace.
So put the necklace there.
Oh, it's so pretty. Can you hear me? Can you hear me? Hear me?
So I think you should get higher.
How about now?
Yeah.
Cool.
Turn the light off.
Where do I start? Is that right? So, hi, I'm Mellissa from the UK, and
the reason that we put this event together is because the majority of us went on a residency called Art Ayatana, which is in Ottawa and run by Alexis Williams. And we're all pretty much alumni from that. And they're doing a exhibition in California, some of the alumni, in the next couple of weeks, or in March. So I guess where I start is what I do, really. So my interests are bacteria, slime, plants, fractals in nature, mushrooms, microscopic stuff, and myself and my invisible self. So, my notes anywhere. Oh, okay. So my first kind of exploration into microbes was to do this project called Microbial Me, which was casting in agar, which is a seaweed-based jelly, which you find in Petri dishes mainly. And I thought I wanted to make this 3D somehow, because it's quite boring in a dish. So I thought, how can I make this look better? So I was really interested in the idea of a face as well, and the self-portrait. So I cast myself, not by myself, because that would be really difficult, and then used different agars to try and bring up different colours and different bacteria. And I collaborated with two scientists in the UK who at first were quite scared of me, because I'm just this artist kind of running down the corridors going, can I make sculptures with agar? And they're like, I don't know. And we tried it and it worked, and it was really amazing, actually. We did this exhibition at the Eden Project in Cornwall in the south of England, and that face has been there for about 2.5 years now. I think it's coming on to 3 years. And it hasn't died yet, and it doesn't look, I mean, to some people that looks gross, but I think it looks quite beautiful. So this was a year later, and it still looks great, right? It looks great. And apparently, I haven't got a photo for now, but apparently it looks amazing as well. Still, so I always also get quite interested in how the work is disposable as well as ever evolving and ever changing, and it makes its own ecosystem as well, because it feeds back on itself to grow more. So after that project, which at the time I found really difficult, I decided to do an even more difficult project for a documentary on the BBC last year, where we made a life-size body sculpture in agar, which sounds amazing, but it was pretty difficult to do. So on one side of the body, we split it in half. One side of the body, there was antibiotics in it. So, I can hear myself now. Can you hear me now? Or was I not? You can hear me? Cool. So one side of the body had antibiotics in it, and then the other side didn't. And we wanted to test and see what the microbiome would react like with that. So on with that, I think the most challenging part of it was actually the casting itself. So my work is really process-based, as I think I like to explain the process of it because I think it adds to the actual artwork. So the casting process, we had to work in reverse, and we had to work in a way where we had to pour the agar in so that it would cast upright, as opposed to a jelly mold where you just turn it around and pop it out. So that was challenging as well, and making it watertight as well, because agar is like thinner than water. And then through this as well, I've got a little video actually. So it took us, we had to pour it at 55 degrees in a really hot room, which the film company made for us, really like a greenhouse basically. It took us 6 hours, and we poured 80 litres into that, and it took about 2 days to set as well. but we had to be careful because obviously, I mean, it would be amazing if someone would invent see-through plaster and rubber, because it would have really helped us in this situation. But yeah, it was really a challenging piece of work to do. Also the quantity side as well, because agar isn't really used in this way. And the scientists who I worked with, they were like, so how many litres of agar do you need for a body? And I was like, oh, I don't know, 30? Like guessing. And they were like, well, we need to find out, we need to know exactly. And I'm just like, okay, 60. And they're like, no, like you need to tell us exactly what it is. So after, because we only had a very short time to do it as well, because we're working with film, they want everything done in a 24-hour gap. But yeah, so after a while, I did a water test, poured loads of water inside it to see if it was watertight. And then I worked out that it was actually like 180 litres. And my collaborator called me and said, Mel, we can't do the project. And I was like, oh, why is that? And he said, well, if it was to fall over and break, 180 litres will have enough bacteria on it to kill someone. And I was like, oh, okay, cool. So after that point, we had to go back, and then we made like a skeleton for the inside of the body mold to reduce it down. So we reduced it down to like half, and then we were able to do it, which was great. And then what else is important with it as well is that the sculpture needs to be, anytime I exhibit living stuff, curators are terrified of me because they're just like, oh, it's alive. But they just need to understand that there's you have to create an environment in it as well. You have, like with the Eden Project one, it's inside a glass, behind a glass wall, and then behind another glass wall. So it's completely out of reach. But with the body one, we got this big kind of glass casing made, and they kind of ignored it needs to be airtight part. So after 4 days of filming this, I'm going to go back to see, of filming this being growing and stuff. They planned 2 weeks to film it. But after 4 days, I came into the lab and I was just like, oh, I can smell it because it stinks, like it's horrible. And I was like, oh, I can smell it. That means we have to decommission it because it's a health and safety risk and we're going to have to shut down the university if it's, etc. So it's a shame because the faces have lasted for years now, and I think a whole body would last probably decades. But again, people are scared of my work, and if I mention the word biohazard, they hang up on me. And yeah, and if my scientist calls, they talk to him normally and it's all fine. But so maybe I just need to pretend I'm a scientist or something. But yeah, so that's been the last couple of years. And then next, I've started working with a immunologist from Manchester University, and she wants to get me a pill camera to swallow so I can film my insides in some way. And then I'm hoping to do a residency as well, researching fungus and soil and how it kind of communicates with itself or with each other to help plants grow. So that's what's next. But yeah, that's me.
So, Robyn Crouch. She said, tell me what I do. I want to connect it to what I do. Mellissa was talking about microbiota, and now we are talking about the chemical. Sure. And we want to do biocode. Yeah. So there's a lot of connection between the two. Just so you know. Hi, everybody. Closer.
What?
It is on.
try and say something a little bit more. Good evening, everybody. Can you hear me now?
Yeah? Good? I'll speak up, how about it?
Hi.
Nice to see you all.
Thanks for coming. So my name is Robyn, and I also like to body attack. Originally, I was inspired by the British artist Anthony Gormley, who did a lot of meditation in India. And his work, he would be inside casts for hours, which is terrifying. Because the first time I was inside a cast, it was for 2 hours. And I got cold, and I had to go to the bathroom, and we've been listening to music, and it stopped.
And I actually saw the claustrophobia close in on me, and it was really scary. But because my face was covered also, so I was in this one, in the fetal position one. So I have a lot of respect for him, and but it's a really interesting process, and it was an experience of metamorphosis for me, because at the time I was going through some really difficult emotional processes, and the plaster bandage, when it first gets put on, is
wet and warm, and as it hardens, it breathes with your body, so as it hardens, it becomes cold and tough, and
I felt like a pupa inside a chrysalis, and then finally when
it was taken off, I felt like a little egg that got hatched. Cool. So,
yeah, my interest started with the body, and then I became interested in, my first interest in
science was in biology as well,
where I just thought of myself as an artist. I
didn't really envision myself as an academic much.
But it was the functioning of
the body system that I found so fascinating.
So body casting sort of led
to interest in more molecular realm. But in the meantime, I became interested in geometry. I was making some tiles based on the platonic solids, which and they are all and so in three-dimensional space, platonic solid is a regular polyhedron, and
all the angles are equal,
and all the sides are equal, all the bases.
And there's only 5 of them,
and we attributed each of the elements, fire, earth, air, water, and ether, to each of the 5 solids. So I was tripping out on that, making 3-dimensional shapes on 2-dimensional surfaces. And so when I came to the University of Montreal, I had come from BC, where I was doing a lot of pottery, because that was what my ceramic constructors focused on. And when I got to Montreal, it was much more sculpture-based, and they weren't really encouraging drawing, so that was a bit of a blow for me. So I had to invent ways to be able to grow as my projects.
So I started
doing the all-around inner-outer treatment of the surfaces, doing multi-fire, multi-layering techniques with transfers. So the ones underneath the glaze are called silkscreen. They're silkscreen tissue transfer depots. And then the ones on top are
water slide transfers.
And so I make those in Photoshop. And so these are all fired 3 times with this firing. 4 times actually. So yeah.
I'm a glutton.
The process. And then I got into body casting again, working with the decals. And
working with the ideas.
This is where the molecules came in, because I became interested in brain function and neurotransmitters. And imagine these little molecules as being they're like little robots. And I see these as like little stories and micro-mythologies as the molecules interact with the forces of nature speaking in geometric shapes. So this is my current work. Continuing with the pottery that I love it. Continuing to explore, getting to know more molecules because it's endless, as I'm sure you know. And what I'm really interested in is
the lock and key mechanisms
between carrier proteins and the molecules that come including them. So that's where I'm going into researching how to actually portray a cell membrane and the molecules crossing the barriers.
Into the cell. So,
and by a man named J.P. Talsek, speaking about the intersection between art and science. And he says, at times the role of the artist is that of observer and critic of our evolving culture. Where scientists seek to answer questions, artists often ask more questions than they answer. Other times, the role of the artist is to explain, illustrate, and educate, often making the invisible visible. So, I like to use the visual art platform to explore the human self as an interface between the invisible and visible worlds. And with that, thank you.
Can everyone hear me all right? We're good? Okay. Perfect. All right. So, hello, everyone. My name is Shavon. It's actually great to be back here. I'm an alumni.
From the University of Toronto. It's interesting to be back here on the context of not being a student.
I am kind of caught in between two worlds with my practice. I studied art history out of Toronto, but I also worked with environmental science as my other background. So I did quite an elaborate hybrid between the two spaces and try to create a practice that kind of talks about my relationships of what I learned within the environmental aspect, whether that be from doing like environmental assessment or through hikes, conversations with faculty. But I also try to bring it back into my actual art practice in a thought-provoking way where I use art more as a tool or a mediator to discuss certain environmental political issues that are going on today. So the first one I'll talk to you about is thirst. This is a series I did in 2016. So I grew my own algae, specifically blue-green algae. I grew it from tap water found in the GTA. My background, I grew up on a farm, so I'm quite familiar with fertilizer runoff, using different types of fertilizer. But until my studies at U of T, I didn't fully understand the repercussions and where everything kind of leads into after our first use. So with this piece, I went to different bodies of water, from Caledon to Brampton to Markham, and directly from Lake Ontario. And I sampled areas that had eutrophication through large amounts of algae blooms on the top of the surface. Now, I'm very aware that blue-green algae is quite toxic. So having this displayed at any gallery, kind of like Mel, I ran into a lot of problems. Not a lot of people wanted to deal with me, especially because with this piece, there is no lids on top of them. So it's a series of Petri-like dishes that have algae growing in each one of them. while there's grow lights on top. So while it's installed in the gallery, it continuously grows. Now, if you hit one of those petri dishes, it will collapse, which is kind of an interesting, I found interesting myself, because that's how waterways work. Lake Ontario is kind of the end process before it goes out and distributed into the oceans. So as these collapse, they'll funnel into each other, connecting everything together. Another series that I worked on is called the Mark series. So this is focusing on the emerald ash borer and ash trees. I started to notice in most communities these orange marks. I knew the emerald ash borer was definitely on the rise. They've definitely moved all through Ontario into parts of Manitoba, but I didn't really understand the impacts, what that would have on a community basis, until I started seeing the local trees be cut down. With this, I started walking over about 8 to 15 kilometers a day, collecting individual photographs of the mark. The mark itself is just spray paint, but I collected about 350 images, which are in the install here. This is at the Latchman Gallery, who I also showed with like Edward Residency and a few other artists like Moira Ness. In total, I have about 1,000 photographs documented. They're not always on display together, and it's an ongoing series. From there, I really started to focus in on, at this point in my science practice, I was working with Monica Havalka at the Mississauga campus, and I was doing an environmental assessment at Scottsdale Farm, and we were looking at invasive species, or at least the debate of invasive species, exotic and native, their values, their effect on biodiversity, their effect on our relationship with the land, and also our effect on how we perceive Canadian icon species. So I realized that this is a topic I had to discuss, and so it fueled a bunch of ongoing series like this one. I had the pleasure of working with Canada's 150th BioBlitz at the Rouge National Park, as well as the Thousand Island National Park in Kingston. With this series, I put up a bunch of QR codes. This is at Thousand Islands, where people could scan the QR code, and from there, it would take them to a virtual reality of where they would be on the trail. From there, it would be a website that would tell you, would give you a list of different images of the plants that you would see, from the flora to the large spruce in the area on the trail. And you could pick which ones would stay and which ones would go, essentially allowing the viewer to be their own curator of the landscape. And what kind of landscape would they idealize and think about as more, why some species have more value over others. From there, I did a piece at Rouge Park. I'm just one agent. This one, I realized when discussing the relationship to humans and the Anthropocene in the environment, that dispersal method was left out in the definition, that humans were not a part of it. We have wind, we have water, we have explosion, which is how these seeds move away from the parent plant. But a lot of the times, humans, especially with the amount of us, were the ones actually being carriers and dispersing them. And because the government was very nice and allowed us to use the parks for free, the national parks, a lot of scientists were actually concerned that the rates of invasive species populations would increase because just the fold of how many people are walking through the parks trying to learn more about our Canadian natural history. So this series I did within a day, end up walking about 20 kilometers. So it was a specific guided tours. I would give 5 kilometers apiece. I would talk about the debate of invasive species, our roles, and I'd target certain ones that we'd find along the trail. In the end, I would ask the participant, the viewer, they could scratch the bottom of their shoe, and I would collect the seeds, among other things, dust, stones, in a communal jar. From there, I haven't quite decided what I'm doing with them yet. They're still sitting there. And the final piece I've been working on, this I did with a chef, Brian Williamson. So trying to kind of bring back the reason why invasive species were brought here, or at least a lot of them, like garlic mustard or wild parsnip, was because they had value to our settlers that came in the 1700s. Now, today, many of those connections to why they were first brought here have been lost. So in an attempt, I've been working with those invasive species directly that are still edible, gathered and harvested from the GTA, and I put them with a piece with the chef, a bunch of appetizers that people could eat during the gallery opening. Thank you.
Tosca Teran. Yeah, you have to put it way up.
Does this, like,
no.
Who can't? You're talking to people.
So, there you go. Okay.
Hi, I'm Tosca Teran.
And I live in Canada. So when I first came to Canada, relocated here in 2001, pretty much around that time I started reading a lot of Ray Kurzweil, The Age of Spiritual Machines, and Nicholas Negroponte's Being Digital. I became really fascinated in smart dust and all these things I was reading about coming out of the media lab at MIT. And myself and two other artists created an installation called Transgenic Morphosis, where we speculated, we looked into the future to look back at this time and then perhaps a little bit farther into the 21st century to see, technology, what were the people of the 20th and 21st century messing around with, and we were just speculating on that. So I created non-invasive implants. I like to take the ideas of small and make them macro so people can maybe better understand or look into that. So I use a lot of gut. I work with metal and a lot of other materials and sensors. So proximity sensors were in there, and there were stereoscopic images, and that's what the one on the left is. These phages, these robotic phages that would be entering the bloodstream. And body mass continued with my, in quotes, non-invasive implants, which is kind of a joke kind of me, because they're very invasive. Using the neck piece is sterling silver and maybe LEDs. And then I guess I should say too, the main materials I really work with are metal and glass. And I love combining the two together. So with Orbis tertius, I took a Barton Orbis tertius and I made like a little museum setting showing transgenic organs that were like maybe human biomechanical mixture. And this is showing that life-size anatomical for the heart.
Can you speak louder, please?
Oh, yeah. Can you hear me?
Yes.
Sorry?
Speak louder.
Okay. I will. Okay. Woohoo!
You don't need to speak louder. You can just put it.
You can like
I
choke
don't
me.
want to choke you.
Okay.
Try now.
And let me look at my notes. So my work explores terrestrial manifestations and conjectures future implications of technology, science, and politics have on us and our environment through a multidisciplinary approach, often combining tactile sculptural forms and audio, and most recently I've been starting to work with sense, creating immersive environments, unnatural history dioramas, and performative wearable structures. My one-of-a-kind jewelry often serve as maquettes and experiments towards larger sculptural work.
One of my many inspirations
has been zoologist Ernest Haeckel.
Haeckel was a champion of Darwin, but he also embraced the pre-Darwinian notion that life formed a series of
Successfully higher forms, with embryos of higher forms recapitulating the lower ones. Haeckel believed that over the course of time,
evolution added new stages to produce life forms. Thus, embryonic
development was actually a record of evolutionary history. The single cell
corresponded to amoeba-like ancestors,
developing eventually into a sea squirt,
fish, and so on. Haeckel, who was adept at packaging
and promoting his ideas,
coined both a name for the process,
the biogenetic law,
as well as a catchy motto,
ontogeny recapitulates the phylogeny. Haeckel was so convinced of his biogenetic law
that he was willing to bend evidence to support it. Haeckel used the following chart to show that we resemble these animals. During human embryonic development, the embryo recapitulates or repeats human evolutionary stages from a fish to a chicken to a dog to a human. Included in his claims are that what Haeckel said was clearly a yolk sac. I think you can see it. From our days as a chicken, a lizard's tail from when we were reptiles, and gill slits from when our ancestors were fish. None of this is true. Nevertheless, biogenetic law became very popular, and Haeckel's illustrations found their way into biology textbooks. If we can say that the world of science is synonymous with truth, and the world of art with that of fiction, I want to tread a middle ground that is unusual and seemingly beyond belief, yet also familiar. So this is from my Unnatural History exhibit that took place at SOFA, New York in 2009. I really was interested in fungus and in particular cordyceps, ones that invade ants or take over moths. There's like hundreds of different types of cordyceps, and they influence what these creatures do. Like for an ant, they generally will climb up to the highest point and embed themselves there, and then the spores grow out of their head. And this to me is incredibly fascinating. Like I want to see that. I want to make that. And then the air we breathe is actually, it started in 2013, and I'm still working on it. Sorry. In 2016, I was awarded a 3-month residency with Ness in Skagesjond, Iceland, where I met phycologist Dr. Bettina Schultz.
I was allowed access to the marine biology lab in Newport to research algae, plankton, blooms, and discuss the marine food
web's ultimate
demise with Dr. Schultz.
My interest in algae grew into an interest in our
microbiome, which started microbial
cloud explorations, which occasionally stress me out. Here's an algae
installation. It's powdered glass. And okay.
So talking about microbial cloud explorations, I just kind of added this. People here in Toronto will totally understand and get the reference with the TTC. But thinking about that, especially on a day like today where you have to take public transit, but I'm interested in how we interact. Like I want to know more, like how is our biome interacting with other organisms? I mean, we're like, what, 10% human or something, that made up of all these different organisms, and how, like, I have cats. How do the cats interact? How do their microbiome interact with my microbiome? How do I interact with plants? So the little animation isn't playing, but my hand has glass bacteria and different things that I've flame worked, and they're moving around. So something that I'm wanting to work towards now is a virtual reality representation with glass 3-dimensional representation. So people can look at these organisms in macro, and with the VR, they can
perhaps enter a generative bacterial environment and see exactly how they might be, how are we with flora and fauna. are we transferring these things to each other?
I'm just an artist, like
I'm not a scientist at all,
so, you know, I don't even know
maybe if any of this is true, but I like to imagine these things and see where they go from there. And that's about it.
the little.
I enjoy it.
How's it going? I imagine you probably can all hear me. I've been told that I'm pretty loud. Is it too loud? I can. All right. Everyone's like, I'm awake. Ah. All right. Oops. So yeah, there I am. Thanks for coming tonight. My name is Julia Krolik. I have a little bit of a weird story, I suppose, in terms of how art came into my life, because as someone who came from Russia with a mom that was all about science, she kind of said, you cannot study art that makes no money. So you can imagine my sentiment, but I love creativity and I am a very creative person, but it was a little bit stifled in kind of this dream of becoming a successful scientist, especially at the time in Russia where being a scientist was the most prized occupation you could have in the tech race. So it was interesting. I kind of resonated with that. But, you know, one thing that I learned is that, you know, if you put 10,000 hours, as they say, into something and you're quite passionate about it and you spend the time and the hard work dedicating your time to that, you do learn something. And so I immigrated to Ottawa, and luckily we had the National Art Gallery and at the time the Contemporary Photography Museum. And so for 5 years, every Thursday from 5 to 8, I went to the National Art Gallery because my mother couldn't stop me. And so I put in my 10,000 hours into the exhibitions, into the art, and I always thought of that space as something that was maybe inaccessible to me as somebody who knew that I was destined for the sciences, but I envied it all. I thought, oh man, this is exciting. And science art was not something that I thought of at the time. And so I ended up doing a bunch of undergrad in science. Then I ended up doing actually a college degree because a colleague of mine said that don't do grad work like everyone else right away. Go do something that's applicable. So I studied medical laboratory science, and I was on placement in Victoria getting my microbiology certification, because MedLab science has 5 disciplines that you become an expert in the lab. There's histology, there is transfusion, there is chemistry, microbiology, and hematology. And so what was interesting to me is the lab I arrived at is a level 3 lab. It has tuberculosis culture. And I thought it was the most fascinating thing, having done rotation at the hospital and seen the actual ward and how long patients are quarantined for, and kind of studying mycobacterium as an organism. When I looked at the slide on the right, there is a slide of TB, and what the bacterium is doing there is called courting. It secretes a certain protein that is very favorable for biofilms. But when I saw this was my first aha moment, because I thought, now that is really beautiful. And I kind of had that aha moment. I thought, but I am so lucky I get to see this, but nobody else gets to see it in this way. This was in 2009, and I thought, I wonder if I could do something that will bring bacteria to people, and maybe it'll bring their environment and their bacteria, not just, you know, bacteria that's clinical. Because in my clinical practice, I was always taught to ignore the environmental bacteria and really focus on clinical pathogens. And so when I came back to Kingston from out west, I said to the college, is it okay if I borrow the micro lab for the summer? And they're like, and this is where I feel actually that I'm in a position of privilege to some extent, because anytime I've ever asked to do anything scientific as a person who is scientifically educated, I always get the green light. And it's interesting for me because I'm not a person that needs to necessarily collaborate with a scientist, because I can be that scientist. But then I also took the steps into being the artist. And so I find myself sometimes thinking about that, because they said, yeah, totally, order what you need, have the lab, go for it, it's your lab. And it was incredible to walk into the place where I learned clinical micro and the scientific method, and to be reproducible and as truthful as possible and take care of people, to kind of just have it to myself to be creative. So what I decided to do, I wanted to show Kingston its bacteria. So I went around and I collected soil samples from all kinds of places in Kingston, the local, sculptures and art installations, to where people walk at the market, to our ferry. I went everywhere. I went up north, and I was curious. I had a question in mind as well. What does the floor look like? And so I spent 2 months doing the project. I grew the plates, and then I thought, at what point will I stop treating this as a science and actually turn it into art? And so staining was where I thought I could take liberties, because I didn't have to classify anything. So I ended up lysing cells. I ended up treating bacteria as my canvas and the staining as my paint. And it was really fascinating to be able to break free from the rigidity of science and to just be creative in this process. So I approached the local gallery, and at the time, of course, I have, a science CV with no art background, and I said, well, do you judge work just on artistic experience, or is it merit in the aesthetic? Because I think these images are great and they should be seen. So I ended up getting a solo show, and then later that was a photographic installation that was geospatial. So as you walked in, you kind of had west and north and east, and so you can space yourself relative to where Kingston is and look at all the locations and see all the bacteria. And then I collaborated with my partner, who's a creative coder, because I think that projection would be really great here, and what kind of a visualizer can we make to visualize the images. And so that was exhibited there at the Propeller Gallery, and then it was also at the Ontario Science Centre on the floor as these big circles, which was really cool. And so that was my brush with bacterial work and bioart, and I did a couple of other smaller projects, but then my science career at that time, I was doing graduate work, and I was studying private well water quality. I was working for the government as a researcher, and I was studying hydrogeology. I was doing geospatial analytics, and really was just looking at groundwater issues in terms of public health. So looking at pathogens and microbial tracers, and looking at different genetic methods that you can trace bacterial contaminants in water. But what I obtained in the process was a really cool data set. And what was interesting is that when I received the data set, I was looking at getting a land use map out of this. And so what you do is you create an algorithm that looks at all of the different colors in the pixels, and then it spits out an approximation of where there is, natural land versus concrete and industrial land, et cetera. And that was going to be used for some spatial analytics. But I looked at it and I said, this is really cool. We should get this out somehow as an art project. I think it'll be a good art project. And again, in collaboration with a partner, I said, well, what can we do to visualize this, to kind of comment on the urbanization process? Because I just found it really fascinating that, and this work is interactive. I'll turn it on in a second, or rather dynamic. I thought, how interesting would it be to kind of showcase the data set itself, but also kind of make a running commentary on an area that you can get into really heavy urbanization and then move out where there has, urbanization hasn't occurred yet, into more of a natural GTA landscape. And so when I called the ministry and I said, I want to use the data for art, there was a really long, uncomfortable silence. What? So then I was like, well, I'm a researcher studying blah, blah, blah. They're like, okay. And I said, but I want to use this for art. But again, that position of privilege in understanding the data set, they felt comfortable in talking to me. Still took some time. 5 people later, I was talking to the guy in charge of the data sets, and I said, look, I can pull free aerial data and make it look like it's the GTA. Let's be real. But I want to use our data. We, as Canada, have invested money into this project. It's beautiful imagery. This is the data that I want to showcase. And so they agreed, and we did. It was first as a film, and it won the Toronto Urban Festival Canadian Urbanism Award, which is really cool. And then it was showcased at the Scotiabank Photography Festival. I'm going to see if I can get the video going so you can see what it looks like. I need permission. Well, I guess you can't see it. I don't even know it. I'm trying. No, I got nothing. Oh well.
You can go online. You can add the.
I'm gonna see if I can.
It's in your corner, right?
I'm just gonna see if I can pull it up as a website and just move the tab over. You need permission to view your own video. Right. Okay, I'm loading it up. I'm just going to move the tab right over and see if that works. Is it being?
It works out.
Okay. So that's what that looks like. So that's actually running a live algorithm. There are a number of tiles that are running. We exhibited it as a physical artwork as well, and it was really fascinating because if you watch this long enough, there will be some tiles that match up, and I won't get into the math of calculating exactly what the occurrence of a double or a triple matches, but at one point we heard people cheer really loudly in the room where this was exhibited. And we're like, what's going on? And they're like, we saw a match! So I thought, wow, okay, this is like a slot machine or something. You want to will it to have the images. But kind of getting aside from the humor aspect of this, which is, definitely there, sometimes, there was a match. See what I mean? But it's interesting how the roads kind of just connect, regardless of whether or not it is a perfect match. We build very, very, very familiar and similar landscapes. So something interesting happened after that. Of course, I kind of got really obsessed with. All right, I have to now close this out. Let's get back to the presentation. Hold on. You know what I'm going to do? I'm going to show you the next work since I know the video isn't going to work. I'm just going to show it to you right out of here. I wonder if it'll let me do that. Let's go back to here and go back to here. Great. Hopefully that loads. It might not, depending on the extensions. Oh well, it's okay. There's an image of it here. I'll go through that. So, of course, going back to what I was doing on the science side and looking at private wells and just groundwater wells in Ontario at the time, I was working with this data set because I had to release several academic scientific publications on this. But I got to know the data set so well that I thought I wanted to do a work that was relevant to water and also relating to, again, the geospatial nature of it. So what's fascinating about this work is that if you look at the top of, and this is a coded work, it's just that particular browser isn't running the extension, the landscape moves down and then you kind of get these bars at the bottom. But at the top, like even here, you can see Wasaga Beach and Barrie, and you're familiar with the geography, but these are just holes in the ground, right? There's no geography here in terms of anything but the holes in Ontario. So there are so many of them, over 600,000 in fact on record, that you actually map Ontario with the holes that are in it. And so depth to water, what it does is as each point comes through, there's a query to a SQL database because I stored them all in a database, and we are pulling some information that the drillers include when they drill a well. And so here we're displaying the depth at which the well was hit, which is the top blue bar, and then the total depth at the bottom, and this is a 100-meter scale. So you actually, at any given point, you see a slice of the aquifer through this visualization. I'm going to try to get rid of this tab so I can just finish off our presentation. There we go. So yeah, that was up to water. I know the video isn't going to work. But I'll just talk about the things that I'm up to now. So I did two things. One is I started an organization called Art the Science, and we have two foci really, and they're in that same vein. I think we're starting to launch artist residencies. We have done some already, and we have one coming up in environmental engineering. But the main focus of the artist residencies, the science artist residency, is to put an artist into a science lab for 2 weeks immersively, teach them the methodology and the science that's in that lab, and then they can disrupt it creatively. It's very important to me as someone that has one foot in the scientific community and seeing what their pains are and seeing how they view science communication, interaction with the public. And then as an artist, seeing the barriers to doing scientific art and how strong those barriers are, and trying to connect with scientists, trying to convince them of access, trying to get in there. So we're working with scientists that are open-minded to this, but also I really want to give the artist those skill sets because I think that will resonate with them for if they learn tissue culture, whatever it is that they learn in that lab, that'll go a long way. And then we're also launching a data artist residency because I absolutely adore data, and there's no shortage of data. Canada, Open Portal, Ontario data, all provincial data sets are there. So we're focusing on creating a data artist residency as well. So rather than having a physical immersive residency, we will facilitate and help the artist gain the digital literacy that they need to work with data sets so that they can do something creative with them. So that's coming up for the science, and then my creative and art studio is called Pixels and Plans, and we do two things. We do communication design for government organizations and helping them with that, but also we make creative projects like Depth to Water. The only other thing, do I have that? No, I don't think I have it up here. That's okay. I'm gonna end there. And I think this is the next slide. Yeah, this is everybody and their social media. So tweet away.
But right now we have, what do
I do with the amulet?
We are calling all the speakers to sit at this table so that you can ask some questions and we can actually have
I guess we're going to need this, so pass it around.
yeah. The way it works is when you and you speak into it. And people were asking questions when you speak up. Because like we have complaints. And we don't have anything that passes around. So who wants to go first? I have a lot of questions, but please go first. You're very welcome. Yes. Samantha and then other lady.
My question was for you when you're showing the microbes and what the integrated they are to magnify sleep. You weren't really sure how much truth there was there.
Yes.
Can you talk a little bit about how important like the accuracy versus the ideas are in science art?
From my perspective,
Yeah, sorry, I need the amulet. It's like I could. I know. As I've gotten more into, I guess, recognizing that a lot of my influences come from science, I'm finding for myself it's important to find what's accurate, what's like fact, what's happening right now. I'm trying to get microbiologists I know to give me access to the lab. Like, talk to my boss. I'll talk to my boss, but not now. Talk to me later. So, but I would like to be able to depict at least as close as possible in the glass and in the VR aspect, which I'm guessing would be like three-dimensional models interacting as close to representative of different bacteria or yeasts and things that are going on. And then I will imagine that's when, like, I guess my fiction with that, like how, if I pet an animal or if I walk by a plant, maybe some of that plant is onto me and I don't I think that's, I don't know though.
Yeah.
If that could be.
It's a cool difference.
Yeah.
So my question could be for anybody, but maybe particularly Shavon and I'm really excited by the potential that this kind of work has for activism and showcasing social justice issues, especially when you have privileged access to a data set and also privileged access to those skills that would allow you to interpret and visualize that data set for a non-expert audience. And so I'm wondering if you've explored that in your work or are thinking about exploring that, aware of other people who might be working with this kind of data to highlight issues that may not be that sexy and bring awareness to it.
But we can both answer. Sure.
I think you'll have more to say about it than I do. I think for me, in terms of art, the science, I'm really focusing more on science communication and just educating the public in general on scientific methods. I feel like there's a lack of that kind of general public understanding, and so it would be really nice of current research. So it's really important for me that it's not about the fundamentals of science research. It's more about, well, what are the researchers doing right now in their lab, and what are they working on? Not so much about how does physics work in general, how does chemistry work, which is also important and amazing, but I feel that it would be really awesome to kind of blend the two. And so that's my main focus right now, rather than so much about activism. It's really about communication of science, and then opening opportunities to artists, because more and more I see how much of an interest is in that practice. So turning it over to the activism.
I think for myself, activism, art activism is very important. Majority of the times, regardless if it's physical art in this sense, where you have an artist that has sculpted something, painted something, or you have someone who's worked with a documentary, I find a lot of people always say, I saw something, and it caused me to think about that discussion, and it started to create a platform where people who don't have a large body of knowledge within that sphere to develop that kind of communication, which I find fantastic. For me in my own practice, I find making art is a labor-intensified process, no matter what version you're going with, but that is a great tool to have discussion. For me, the emotional response is probably the most important. We can give stats to people, no offense, all day long, but that doesn't create always social change. Some people it does, but unfortunately, as humans, or not necessarily unfortunately, but fortunately, as humans, our emotional response is how we trigger whether or not we're going to do something again, because how it made us feel. And I find art is a fantastic tool to create that.
Other questions?
Yes. This is for Tosca. I noticed that one of your sort of green glass notes was very laid out by Haeckel's pictures of the creative area. So, one of the great admirers of him was a famous guy named Darcy Wentworth Thompson. I don't know. He is the.
Sorry.
Last year was the 100th anniversary of Darcy Wentworth Thompson's famous book called On Growth and Form, which was published in 1917. And in Britain, they had a whole Darcy Wentworth Thompson year, and there were special programs all over Britain about him. And he was quite influenced by Haeckel.
Haeckel, yeah.
Haeckel, and yeah, and there's the glass
artist, and I've wondered about this, named the Velasquez, who started first making glass eyes, and then I think they continued with that, but it was really
popular, right, to
gather menagerie, like the Victorian times, and you couldn't really keep jellyfish, or people didn't have, and they'd stay well-defined, or whatever horrible things happened to them. And then Velasquez started
creating these models. Harvard has a lot of
their plants. The Corning Museum has some of their vertebrates and things like that. And I believe there's a huge collection of the National History Museum of London.
It's amazing, but they look like Haeckel's drawings. So I've asked historians, did they look at his work, or was he looking at their work and drawing, doing the engravings? Anyways, I've just.
So everybody knows that Ireland won the. Wentworth Thompson was a proponent of the idea that things organize themselves into forms, because those were the correct form. And in particular, somebody, I think Pock had a picture of the famous cube, soap bubble cube with the small cube in the center. I don't know if you realize what that is, but it's a soap bubble. You realize that soap bubbles organize themselves like radiolaria and so on and so on. I wonder if you're all aware of this whole connection between. We have a large collection, for example, of mathematical demonstration plaster things, and they just look like radiolaria. And right outside here in the lobby is a projection of an 11-dimensional holotope, which is hanging from the ceiling, and it looks like a cluster of cells. So there's actually a lot of
Yeah, I'm going to get that name.
What's on the podcast?
So you're all bio-oriented, but I'm giving you another idea, which is math, all math. If you go down into the guts of it, you'll find that when we're constantly the connection between eco and math.
Okay.
Question.
Yeah.
Specific question to Julia regarding your artist in residence. I'm wondering what your experience has been like with researchers when you're approaching them, and what kind of buy-in you're getting. Like, do you have people coming to you? Do you have interest, hesitations? Just like overall, what do you see in science globally?
So I'm not getting hesitation, but I think because the conversation is with my colleagues from the science world, right? So these are folks I've already collaborated with on academic publications, so they're not people that are necessarily strangers to me that I've started piloting this with the intention that eventually, it will get picked up by folks that are maybe not so open to it. In a separate project, I did something called customer discovery, where if you have a tool or an offering, you phone as many people as you can, and you disguise that you're finding out if they would buy your product, but you ask them important questions. And so I ended up doing that, and I spoke to many researchers. And what I found out that there are two types of researchers, and I'm going to totally generalize here, but this is from obviously a sample size of 100 people. What was interesting is that either you have an individual that is so caught up in their work that absolutely has zero need for knowledge translation or talking to the public at all. And so they're like, hey man, I'm working on a molecule. I don't care about the people. Like, I'm focused. I'm going to make this great discovery. That's amazing. They are that focused. They're that passionate. It's like, life work. And then you have a different type of researcher in the science world that, all of their experimental farms are on local farmland, and they go to the town hall meeting and they talk about, the importance of that research, and they need that support from the farmers. Or you have a lot of funding from, different foundations. So there is a need to communicate the research, and those are the very interested researchers that are like, I have a personal interest in getting that out, and what, better way of doing that than to explore a novel method by inviting a creative in. And also, I think it's a very mutualistic relationship for the immersive residency, because you have somebody who can come in and disrupt all kinds of processes and ask all kinds of questions that will cause that researcher to pause and say, I didn't think about that myself, because, that's not why I'm doing this. And then in return, of course, for the artist, they get a whole different medium if they want to play with it. So, so the one at Queen's is coming up. We're working with one at Laurentian University and at Western, and I have an email box full of stuff. So it's in the works. It's just slow. Because as we're doing these, we're also doing program evaluation, and so we want to really evaluate the merit and improve the residencies as we grow forward with them. So it's important that, to me anyway, that they become better as we go, so that. And also, in speaking of the academic language, having some kind of academic record and being responsible for what we're doing, I think will help us when we are talking to different potential collaborators, because they're not going to want to believe that it works. They're going to want to see that it does. And coming from a quantitative background, sometimes stats is important. In this case, it would be. You haven't said anything. You should. Hi.
Actually, I'd like to bring her into the conversation now, because since we started talking about access to science and the way in which we are considering art in the service of science as opposed to science and arts collaborating equally, I would like to bring up this thing and relate to your own experience dealing with science. Like, I know that Julia is already a scientist, and she was telling me that, and she was actually saying that she has somehow privileged access to, of course, to scientists. And also, you are also more listened to and legitimated because you know the language, etc. But for example, for Mellissa, she seemed to have had like more problems. And Tosca seems to say that she has, like, she's been, like, having scientists dragging their feet, giving her access to things. So I'm not so interested in knowing But I'm kind of interested in knowing why and why you think this is happening. Like if it's a question of language or it's a question of approach. But what I'm interested in hearing from you is if you think that we should all massage our art and turn it into a big science communication project, or if we should actually try to go like Don Quixote and against the mills and try to actually change a little bit the environment and try to break these disciplinary silos that are actually endangering research in some way. Because as we know, it's disciplinary silos that prevent us from doing better research. So I'm asking you, all of you. So now Mellissa has to talk.
I mean, can everyone hear me? It feels weird. I feel like I'm meant to eat it. So, I mean, it took quite a few years to be, I guess, accepted in the labs because everyone hated me for a good few years. Because some PhD students would be like, that's not science. What are you doing in here? And I'm just like, well, I'm trying to explore something the same as you. But also the smell as well of the work that I made didn't make me very popular either. My long-term collaborator, Mark Clements, we've been collaborating for about maybe 6 years. And he said to me recently it took him about 1.5 to 2 years to actually get used to me. Because it is that language, I guess, like science language versus the art language. It's very different, and you don't really realize it until you say something a bit weird. Like I heard some students in a lab saying, "Oh, I'm going to transform this bacteria," and I was like, "Ooh, into what?" And they were just like laughing at me, and I was just like, "What?" Like so, and it's just all about. I think I know. But also it's I think because scientists in a lab are so used to being around scientists and not artists, and I think knowledge is power, and I think that if you ask, say, you don't know what a word means, and most people, I don't know if it's a British thing, but most people just pretend like they know what you're talking about. But I'm just like, okay, what does that mean? What does this mean? What does this mean? So I guess people, I think they find it, scientists find it very ego-boosting, I guess, because they're like, I'm going to explain what I'm doing, and I think that they really appreciate what I did eventually. But did I answer the question?
No, I just wanted to relate it to your experience, which you did, but also I wanted to talk about like access and whether
I think
learn the language and just say, okay, so my work is just totally about science communication. I don't care about like arts. I don't care about other meanings.
Or whether they should be separate.
I'm trying to change. I'm trying to change things. I'm trying to.
I don't know if we should change things. I think it should be more, because I think it's quite unique still, and I think that's why it's amazing. Yeah, merge the two fields, sure, but also it's that uncomfortableness with the art and science that is quite endearing as well. So I don't know if that's my experience anyway. Yeah, and also like, yeah, I think it's interesting. The interactions are interesting. Like me and my collaborator kind of study each other's interactions as well, because like he says that since working with me, his science has been better because he's actually really thinking about it properly rather than doing the whole like textbook stuff. And is that he says it's opened his mind up to more kind of topics and visually representing things. Because the reason I do what I do is because
I'm trying to communicate and get people to relate to what I make, because everyone has a face and they can relate to it. I think I'm done.
I think there's room for both. I think that you can have a whole artistic practice around science communication if that's what you want it to do, or you can have a science-inspired practice where you take science to a whole other level. I don't know why, I think there's room for having that. In terms of access, I think that one of the things that we want to do at the Science, for instance, is we're developing 3 wet lab courses. One of the things that bugs me and keeps me up at night is the fact that if I wanted to learn how to oil paint or how to do photography or whatever artistic thing that I didn't get to do because I don't have a BFA, but I wanted to just focus on that one thing, that's accessible to me. I can find a course somewhere and I can master that skill if I chose to do that. I may not be the greatest oil painter in the world, but I can try that. It's open. And science isn't like that. And you don't need a Bachelor of Science to learn how to just grow bacteria, for instance, if that's the only skill you wanted to master. So we're hoping to release 3 programs that artists can take that we can actually vouch for them and say, okay, you're accredited, you actually can do this, so that when you approach a scientist and you say, you know what, these are my competent things that I can do so I can come into your lab, and you won't be so scared about me coming in there.
But isn't that great though?
They're scared about,
to some.
I think being out of your comfort zone
is really important. And I think that's what makes art and science amazing, is because both of you kind of don't know what you're doing.
Yeah.
You go together and you
don't know what you're doing together.
But then you end up making something great, and then
that's research. That's fun.
Yeah, but I think you can still do that if you have some fundamental skills in the method, right? I'm not talking, even for yours, you still have to learn how to, make agar, for instance, or that kind of thing. So if you already have that, then it just frees you up to actually create. And I think that's what I'm talking about. It's just the very basic fundamental things that...
Sorry, but then what's the point in
collaborating with scientists who know science?
Well, I think that then it really becomes about what you want to do once you have that skill set in that lab, right? So if you want to do something about their scientific work, then you can, but you don't have to do that, right?
But then, maybe you kind of
in a way, in that, you go and you had no idea, there's lingo with everything, right? Even in all of the different fields of art, in everything, my metal work has a certain lingo, and the glass has its own lingo, textiles, working with all the different things. Yeah. I think it's a language.
For me, definitely. I don't know. Well, yes,
the correct scientific.
Yeah. My point is that I can learn body cast a lot easier than I can learn how to sequence genomes
right now, and I would love to open that up. I just don't think it's fair.
I know how to write.
Scientists, I can tell you that one thing that will get you more access is to do your homework. When you talk to scientists and you want to get their attention and you seem to know nothing, then they'll say, that's too much time. I have too much time. I've spent too much time bringing this person up to speed. So if you're already kind of half up to speed and you can kind of prove to the person you're talking to that you've read the Wikipedia pages appropriate to that.
Well, and that goes back to my point, as a scientist talking about it, it's landing on my doorstep and it's about exploration. It's exactly what you resonated. If someone's emailing me and they're like, hey, I actually know a little bit about PCR, I know a little bit about whatever it is, then it's going to be a completely different tune than teach me, I don't know anything.
Yeah, right.
Which unfortunately is, I'm not saying it's a good thing, but that's what's happening, right? Am I going to throw my postdoc on this? What am I going to do?
I think loads of scientists are really desperate though to not do science.
Well,
yeah, scientists I work with are so do something. They want to get out of that. So I think it's a mixed bag. I didn't go in, I didn't know anything about it at all, bacteria or anything like that. And I went in completely blind, and it's been...
It's so charming.
You were going to say something. Sorry. That's okay.
I'll just add one thing. We are talking about 2 separate entities, but there's also a third, and that is actually the physical environment itself. And one of the large criticisms going on with environmental art or just bioart specifically is our impacts that we're having. So we're either speeding up an issue by doing these processes. I know for myself, collecting all these invasive plant seeds, well, I can't just return them back. Now I have the burden of carrying these things around, and it's actually, they're in my studio. So it becomes a whole process to consider when you're making art in this way, and not just for the integrity of the piece or the integrity of the concept that you're talking about. but is it truly worth the impact that you're having on a long-term scale? Yeah.
What happened to the big body? Yeah, so basically, that was actually a really frustrating point of the project because it actually goes back to your point of training artists. So I wasn't trained to dispose of that amount of agar, so I had to watch through a glass window 3 scientists destroying my work and putting it in an autoclave and incinerating it and all of that. So that was really frustrating. But also, we weren't allowed to film it. It was forbidden. Like the scientists, they're like, nope, I don't want to get involved in that. I don't want to be named. Like it's like they're happy to put all of it on me, but they're scared because it's quite. I think science reputations are quite easily injured, I guess. But artists, like, they are as well, but it's kind of, it's different, isn't it?
Yeah. And speaking of which, for those people who were going to see some stuff growing on local and foreign currency, it's actually growing right now in one of the cabinets in the Kotler Student Center.
You didn't ask anybody's permission.
What I'm doing, they're just giving it a cabinet. But yeah, they're just a little bit stinky. If you go very close, you can actually smell the cheese.
Just between Bayen and Kofler, there's a kind of hollow, and it's near the career center on the left. It's closed now, probably. But.
The artwork is by Ken Rinaldo. He's being installed in several places. To here in Toronto, and it's now there. And there's other artworks that you can see, including artwork by Jennifer Willett, who's here. So, who is from Windsor.
About the man and the faces.
You're being recorded? Oh, just...
Okay, sorry. So I see it as like the death, like totally going through like the death process. At least that's what I kind of thought it was about. So it's really curious to me that these faces are lasting. Like I would just think they would liquefy or something. I don't, like get eaten until it's gone and just a little dry dust. They're mold or something.
So the first faces I did were in a small gallery in London, and we were kind of amateurs then and didn't really know about stealing or that. So I came into the gallery one day and the gallery owner was like, "What does that smell?" And I was like, "Go get your lunch and the smell will be gone when you get back." And basically the seal wasn't done properly, but it's bacteria that grows everywhere all the time, and it's not harmful. We just have to treat it that way. Yeah, they just live. I see it as a living artwork. They literally look like death masks and like like things in a coffin and stuff. Like on the filming day, someone said to him, "Oh, it looks like." Oh, he said he was like, "Oh, it reminds me of my father when he died." And then someone else was like, "Well, it's going to look like that in a couple of days." It was a bit like, "Ooh, God." So, did I answer your question? Yeah.
Because it is, we have all these things that we keep in check while we're living,
and then when we die, things totally to go like woohoo. Yeah, we take over. Yeah, so these wouldn't have that control system. I'm guessing they would just have doing.
Places where the little bit of air came into so if there's air coming in, they shrink down, but then the features still stay the same. It's really interesting, and it just causes condensation, so that causes the ecosystem and the feeding back and all of that. But if air comes in, because agar is made of mainly water, if air comes in, it will just shrink down, but it still keeps its its detail, which is really interesting. But I'm really sad that that body is not here anymore, because it would have still been doing stuff. But as I said, galleries are too scared.
But wasn't it a possible pathogen?
Something that I really love about your project, coming from the micro side of it, when I first saw the faces, I thought it's interesting because even though the microbial flora that you're displaying is not pathogenic, what you're doing is you're concentrating it, right? Yeah. So in heavy concentrations like that, that is pathogenic just by virtue of how much you have of an organism there. You don't know what that could do. So it's very interesting, especially like for the body. You're like, wow, that is a lot of bacteria, right? To think about the quantity. Yeah. When you only grow a little bit and then you get rid of the rest when you're doing ID work or any kind of microbial work, to see that amplified like that in real life is very interesting.
This is for Julia. I'm just curious what tools you're using for your data science pieces. So you said, like you mentioned, MySQL.
Yeah. So they're all web-based HTML5 visualizations. Some of them are D3, some of them are Canvas, depending. I'm sure you're familiar with both since you're asking the question. So different environments depending on what we want to visualize. If it's interactive-based and vector rendered for clear display of graphics, or if we want to display something that's a little more pixel on the pixel side of things, so we will use Canvas for that. Just really depends on what the ultimate goal of the data viz is. We'll find the tool to do it. Sometimes not everything is available, but we prefer to keep it in web browser because it's just really nice to have it on the website as documented that way. And then we capture videos if we want to present it in different places, because when you want to go to the Toronto Urban Film Fest, they want to play an algorithm, they want a movie of it. So.
Did you learn the code for all this, or you collaborate with people?
I know some of it, and I collaborate with my partner, Owen Fernley, who is a creative coder. So from working with him now for almost a decade, I can do a lot of the work, but I do a lot of the design work and the editing as well for it. So if we have 1,200 images to process, I would be doing that and then handing them to him. If it gets laborious, then I will join on the front end. But JavaScript is his friend, not mine. It has actually taught me something incredible as a side note from working with a coder for that long, and as a designer, and how much of a disconnect there could be between the two. And I find that that's really interesting to suggest meaningful things once I understood what the limitations of programming are for real. It gave me a different landscape when I think about designing things. Because otherwise it's just like, why can't that be that way? Yeah, it's unique.
Wonderful, thank you so much. I have a little anecdote and a question. I graduated from Concordia with a thesis about interspecies collaborations, and I took that to Agriculture Canada and got a job in an entomology lab. Did tiny dissections in my visual community that I learned in art history. Became really important in that lab. So there are lots of little ways to get into labs and whatnot. But my question is kind of more for Julia and Shavon. Working with non-human organisms, can you talk about the unexpected, the surprise, the agency that you, of those non-living things, and how maybe it rerouted your work, or can you just in general talk about that experience? Actually, not Julia, Mellissa. Mellissa. Yeah, sorry.
Oh, I stopped listening when you
said.
What did you say? Sorry.
Talk about the working with something that has its own
life. It's kind of like magic, I guess. Like, it's really unexpected, and that's what I really loved about it, is because every time I'd go into a lab, I'd be like, ooh, what's going to happen today? And it would be completely not what I thought it would be. And then, like, working with slime mold as well is, like, really unpredictable, and it grows so fast, and it escaped all over my studio at one point. And it's just it's really irritating, because you're like, oh, I made all the right barriers for it. But it's also a challenge to learn from the organism as well. But you can never learn because it's so random. And each time you think you've got it, you don't.
So just to that.
I find that people who work with non-human organisms are very open to chats.
Yeah, because that's why I think one of the reasons why I do it is because it's really surprising and really, magical, I guess.
If you're referring to the microbiota project organisms, my only comment to that one was that I came from the control side of knowing exactly what to do with them and control them. So it was about letting go for me, and seeing the fact that I'm going to like not select for a certain organism and just try to pick out as much as I can out of the soil and out of the environment, which was completely unnatural to me coming from a clin background, being like, oh wow, everything is okay. I can actually include it all.
And being more open, like not having.
Yeah, it blew my mind. It was such a transformation to also be in the very lab that I learned to be so rigid, to completely open that up, and yeah, it was really neat. And also going from human pathogens and human bacteria to environmental bacteria and exploring that as well, because I haven't looked at soil and I haven't looked at, at microorganisms everywhere, but I didn't think about, putting a lens on the ground and really looking deeper, so.
I think at the beginning I was really excited working with living organisms, but then I started to kind of question, like, what am I doing? Because I work in conservation and restoration ecology, and I'm like, I go to work every day trying to stop this from happening, and here I'm growing it at home. There's something wrong with that. So I think with my practice, it kind of led me to step back. I'm like, there's got to be another way I can talk about this and be engaging with the community, with the viewer, participant, whatever word you want to give them, without kind of furthering that process. So I think I've really become conscious of my impact. I mean, my ecological footprint is not going to analyze me growing 6 tanks of algae. It's not going to be part of it. But it would be interesting to more see it from more of like a digital scale and see if that has a better effect than just working directly with the actual organism. I'm just not ready for that extra burden right now.
I want to say I really enjoyed all of your presentations. It's a really diverse body of work, and it's really invested practice. Enjoying about this is watching all of you interact with one another. And I was just wondering if you could tell us a little bit about the residency that you did, what that experience was like, how you may or may not have agreed with that experience, and like, I don't know, what it was like. And then, because also now you're obviously reconnecting, so what were the results of that? That you're bringing this really like mad cat and people putting back together. Cool.
So Mellissa,
Shavon, and I were at the Residency called where we studied
nocturnal animals, and we made a fat detector the first day. We got to solder a little Arduino resistors and things, which is really fun. But basically, we all just really hit it off. There were 2 other women who couldn't make it, and Alexis herself. And we just had fun. And so it was basically like being at summer camp for adult art nerds. Which is really great.
Yeah. It is like an incredible camp.
I did the infinitesimal last September, and that was amazing. We visited different labs, Carleton and University of
Ottawa and
the Herbarium
at the Nature Museum. It was amazing. It's just every day you're doing something and you're learning these new things and foraging for mushrooms. Like, it's amazing. I highly recommend it. Highly recommend it. And I think actually Alexi's starting to, or she's thinking about integrating into it somehow, because right now it's not really so much about producing work when you're there. It's just taking in all this information and research. She's wanting to create a component, like maybe how many days after you've done this that you have time to maybe create or do something together, maybe some sort of group creative explorations or something. Because you just go home and your mind's exploding. Everything that you've just learned. It's amazing.
Well, it's also a great opportunity for a group of artists and like bioartists and researchers all to come together on the same platform. There's always that awkward moment or two when they're first meeting us, being like, "We got a bunch of artists here. How much do they know? How bare bones minimum do I have to explain this? Do I need to do the ABCs?" And as soon as we're past that awkward stage, it's a fantastic experience. What is the bird guy's name? Bernie? Bernie? Oh my goodness, he was fantastic. He didn't even, he just loved birds. Anyway, took us out.
He was an accountant by day and a bird
speaker by night.
And owls, yes. He took us out and we were looking at all different types of birds from wetland habitat, and then eventually, I think it was gotta be close to midnight at that point, right? And he has this amazing bird call for barn owls. And they love him. Anyways, he responded, and they had a nice little conversation going back and forth. It was such an experience I just couldn't repeat.
Although Alexis is a dear friend of mine, I have not attended a residency. I should, and I will at some point. But yeah, I've heard incredible things about them. So.
Yeah, I actually went on the residency twice. So I went in 2015 and on biophilia, which is exploration of art and biology. And then it was just such an amazing week. Like I was hiking in Gatineau Park in Ottawa and collecting mushrooms, like really kind of looking at all of these specimens, being inspired every single day. It's really intense, but it's really amazing as well. And then after that week, I was like, I have to go again. I just have to do it. And then this time just doing like something nocturnal, something that's I'm not, like, it's not kind of in my pipeline of interest, but it's something, the reason I like the residencies is because they give you a taste of something that you might develop later on. And I like the fact that it's a research-based residency and it's not, you don't have to do a final piece. I did a final piece a year later from the first one. So yeah, it's just incredible, and yeah, it's just full of art nerds. So you have to fund it yourself because it's just it's Alexis and her mum, and she's the cook, and then Alexis takes us out in her car and just takes us everywhere, and it's just like a really amazing opportunity to be with a small group of people as well. You can obviously go to your government bodies and apply for funding for things, which is what I did both times. I did a Kickstarter actually the first time, but there are ways that you can do it. But at the same time, like, I think we were speaking about it, how there's not a lot of art science kind of funding available here, and there's loads in the UK. There's loads of opportunities. So I find it really weird because all the people I've met who are interested in art science that I really get on with all from Canada, so I don't get why it's not big here. I hope that they will be.
With the leading edge, that's why.
Yeah, actually growing.
Yeah, I don't know why
Yeah, all of the art to science residencies will be funded. We're working on that. That's But.
It's a huge grant, like 150,000 or something over so many years, and you
get to collaborate with the scientist of your choice. I remember my colleague Lily Young had received it. I think it was it's through the Ontario Arts Council, but I don't know. I haven't seen it. I think no. Well, it was a great thing when it happened, but maybe it will change with some advocacy. Because I know that some women from, this is like something else in a way, but from the City of Craft that used to exist, really started sitting on the boards and talking to the arts councils, and they were the ones responsible for getting the craft project and development grant to happen. So, just saying.
I just know with my experience working with the provincial government and the national government for BioArt, or BioBlitz, sorry, we got an opportunity to go around to the different parks, and we had researchers kind of guide us around. And we had a curator that was kind of our mediator between us and the parks and making sure all like the bells and whistles were kind of checked off. But what they expected from us was to create paintings, like the group of 7. So they would take us, especially at like Thousand Islands, like, okay, here's your little boat tour. So what island would you like to paint from? And make sure, if I see any more of those red Muskoka chairs, I'm going to chuck them. And they're plastic. Let's not even start there. Anyways, so the Muskoka chair is where the parks designate the most aesthetic view of the park and why you should come and experience that park. So that's where they took all the artists to make sure they knew that this is the best place. Well, nature isn't always beautiful. Art isn't always beautiful. And so it's a very interesting correspondence going back and forth.
Red flag.
Yes, they're hideous.
Yeah, in terms of funding, I was just going to say that for Art the Science, the residencies will be funded. We're working on that. And for us, we're looking at the science community to fund them rather than the arts. I feel like the arts already need to fund arts. And so I've been talking to many different people and potential partners, etc. That is my current plight, but I believe that the residencies need to be funded for sure. At least for Art the Science, we're not. But I wanted to ask Jennifer to throw back at you. You have a brand new, amazing, incredible lab art space. Will you have residencies in that?
The camping experience, because I hosted a big art science camp at the Rec Center a number of years ago, and it was awful. Like artists like hurting cats. I find it very difficult camping with coordinating artists.
But also how many people we were?
We had total
20. We had 6.
Like I slept in the trunk of my car. Residencies is that from my experience, I've done a lot of residencies. In particular, I remember working at Symbiotic in Australia, and Oran and Net put 100 hours in every project that came through, and trying to negotiate with the artists and the scientists and whatever misunderstandings they had, and the money and the blah blah blah. And I don't have the time for that. So right now, our lab it's amazing. It's a BSL Level 2 lab, and it's a multimedia theater that is also a functional lab. So live audiences can view what happens at the last wall. But what we do is in 2 ways I work with outsiders. With undergraduate students as well as graduate students who can come and do their research in the lab and collaborate with me or do their own projects. And then the other thing that we do is we are hosting not necessarily research residencies, but production events. So we have our first production event starting in March with a choreographer coming to Copenhagen to dry run some projects where people interact with organisms. And so in that instance, like she's already done all her research, she's got her mealworm, she knows what she's doing, and then we can deal with things like how do you stage that, how do you light that, how do you record that, how do you present that to an audience. So that's going to be our specialization, is that like that back end and the final public interface of BioArt. And in that instance, I think I can handle listening for artists.
You're going to hurt cats. Don't try to do too many cats.
It can be very emotional.
Speaking of funding and stuff, over the past 5 years, has your funding sources and resources been most efficient? Has it changed? Like you mentioned case study, what are the sort of sources that primarily make up?
That's a question that changes every year. I think it's like it really depends what the year is, what kind of year it is, like what you're applying for, because there's so in the UK anyway, there's so many people going for the same thing. I've got
From November to now, I've had rejections daily.
And like
I have to grow this philosophically thick skin,
whatever, like it's human, it's horrible.
But it really varies, and you just have to keep on keeping on. Like.
So I did end up quitting my day job as a scientific researcher, and that's fine. Aside from doing art and science stuff, which does not pay me money, I do it because I love art and science. Pixels and Plans does. Aside from being an art studio, it's also a graphic design studio. So we do data viz projects, anything like that for government organizations, and so that's a bit more innovative and allows me to also scratch and create a dish because I love entering a project at the early stage as someone with quantitative background, but then taking it all the way to like dissemination level, communication level, and branding. So I found my niche there, and that allows me then to free up some time to do art and science and create a project.
The other thing is what stage you are as an emerging artist or a practicing artist. There's different grants available to you depending on where you are in your practice. So if you're only 2 years out from your undergrad, you're not eligible for many grants and scholarships. It's make or break it. It's a starving artist. As soon as you break that 2-year period, your potential up to about $1,000 to $5,000 worth for that year till you're within your 5-year radius. Once you surpass that, then you start getting more funding.
I did a Kickstarter.
Yeah. Sorry?
The Art Science Salon has run workshops a few times. We haven't had one. We've had 2 or 3 workshops over in the physics building. And so maybe we should have another one. I don't know. And maybe you want to say something about the workshop that you're doing tomorrow?
Yeah. Well,
talk about that.
Bringing the map. Yeah.
I just gave
I was just, I apply for grants, and then if I get it, it's like I won the lottery. I have done Kickstarter, like I did a Kickstarter to do my first residency in Iceland, and that, it felt very strange actually. I did feel strange doing that, but I'm making artwork for people and things like that. So yeah, it's tricky. I teach classes too. I have postcards over there. My studio's over on Harvard Street, and that, can help, like, definitely if I'm, that's rent and residencies.
Yeah, I still have day jobs to fund myself. I'm still learning these funding grant application processes.
No, they're kind of terrifying. They bring up all the deepest self-doubt.
Or the greatest level of bullshit that you could muster.
Well, that too, right? They kind of go hand in hand. Exactly. Yeah, so it's quite a process of facing yourself and dealing with that stuff, which can be fascinating if we can take a step back from feeling like shit about it.
Right. That made me think, how do you summarize everything you do in 12 words? I've worked with an editor before to help me do that, because it's like 500 words, 100 words, 50 words, 25, and I'm like, are you kidding me?
Hands-on thing.
Yeah. So
you want to talk about that?
Yeah. So we're running some workshops tomorrow. I'm doing a Design My Microbiome workshop, so I'm bringing loads of body molds of the most intimate parts of me, as well as things that I've made up, and getting people to make their own agar sculptures, and also like a portrait, microbial portrait workshop as well. And then
I'm gonna teach people how to paint using biological stains. So we're gonna just work with onion cells, because there's a nice monolayer you can get, and so we've got some fun stains that we can play with, and then look at them on the microscope, maybe take some fun photos.
And I'm doing a cooking class, essentially. So make sure you're hungry if you decide to come. And working with invasive plant species. I don't want to tell too much because it'll take the excitement away.
Sorry, to say that sold out tomorrow? It is sold out, unfortunately. There's already 30 people, and they haven't taken more. Sorry. It went, okay, so this is one thing. But this is already the
second workshop that we are offering, and we are always offering it at the Hack Lab, which is this independent laboratory in a space in Carbondale. It's a level 1 laboratory, so we're really limited to doing this. But every time we go out and we put out one of these workshops, they sell out in like 4 hours. No matter how many spots we have. Like the first spot, because it was about CRISPR-Cas9, and we had to purchase a kit, which cost us some money because there's an exchange between the US currency and Canadian currency, and we are independent. We only took in 15 people, and actually Tosca was among us. So, and the second one, we are very fortunate to have the artists, and we have space for 30 people because we don't have all these requirements, and we have all the material there. But again, we would have loved for Julia to do something more complex. She wants to do something on bacteria, but she can't because it's a level 1 type of lab, and it's kind of limited. So we would really love to have people who have labs opening up their labs to do these workshops. At UFT, the problem is they're crowded. Nobody will open the lab for us because there's already too many students working in the labs. So we have these independent places, which is it's kind of like we hope that we can grow them with expertise, with people giving us access to the med store so that we can get more material, because right now I'm using my own account to it, because they can't, they don't have access to it. So like these kind of things, right? So I think it's a huge work of collaboration that we are embarking in, and like the residencies that Julia is trying to put together. So that it's something that is growing, but like it's a very, very time-consuming, but something is happening. So we will do more workshops, and if somebody wants to volunteer, we have a level 1 lab that is available. So any other question? It's already 7:58, so unless anybody has any question or any final remarks from our fantastic speakers.
Thanks for sharing your time with us and coming. Thank you.